So welcome everybody. Thanks for joining us for our 1 o'clock class. We're happy to have you with us either in our Zoom meeting, on our YouTube live stream, or watching on Kingdom Access Television or your local cable affiliate. My name's Drew Bush and I'm the Director of Programs at the Fairbanks Museum and Planetarium. In just a second, I'll turn it over to our excellent science educator, Bobby Farlice-Rubio, who you may know. A few quick notes for you. If you're in Zoom, you can chat with us using the chat dialog button at the bottom, and you'll see a few welcoming chats in there for me. You can also ask us questions on our Q&A. So that's the button just to the left of the Q&A, and you can ask them anonymously in there as well. If you're on YouTube or perhaps watching this later on cable access, we just wanted to let you know that you can email us your questions either before class or during class, and the email address for that is dbush@fairbanksmuseum.org. So that's my email address. So thank you again, everybody, for joining us, and I will turn things over to Bobby now.
Well, hello, everyone out there. I'm Bobby Farlice-Rubio, and I'm here to talk to you about genes and mutants and GMOs. So basically, this class is going to hopefully introduce you to how genetics works, meaning the study of inheritance through your cells, what we call DNA, our genes, or like that character, the fox in Zootopia said, "You're done," which of course is not the correct pronunciation. So I don't want to get into too much of the technical stuff. You know, DNA itself means deoxyribonucleic acid. And that sounds complicated, and that might make you want to turn off your Zoom stream right away, but don't, because I want to break things down in a way that makes more sense. I want to turn this molecule that looks like a ladder into an idea that you can hopefully understand. So we have to understand the cell before we can understand DNA, and we have to understand how life needs cells to survive. So let me go give you a video from a really cool textbook. I'm going to switch now to a textbook called Life on Earth by the great biologist E.O. Wilson. And in this book, I highly recommend it. It's available for free if you have Apple's iBooks app. And this is a college level textbook that talks about how your cells work. And they have this amazing video of DNA, and it has animation that I wish I could have seen when I was a young person first learn about this. So I'm going to let that go.
DNA in each one of your cells is just 2 nanometers across, but around 30 to 40 million nanometers long. To keep DNA organized and regulate access to the genetic code, it is wrapped around structural proteins to form nucleosomes, which are then further organized into fibers called chromatin. The chromatin fibers are packaged into gigantic molecular structures called chromosomes. We are now pulling away from a chromosome, out through a nuclear pore, which is a gateway through the nuclear membrane.
The nucleus.
We are zooming back to reveal a cell about to undergo mitosis.
Now, mitosis is when 2 cells reproduce, making equal copies of each other.
The nuclear membrane breaks down, releasing the chromosomes. The chromosomes are arranged in the middle of the cell before being pulled apart, with identical sets of DNA going to each side. The cell then splits down the middle, creating 2 new daughter cells.
Okay, folks, I know there's a lot of terminology in there that you may not have heard before, but I even have a model here of one of those nucleosomes that they talked about. See the DNA wrapped around this like a spindle, like your garden hose wrapped around the reel outside by your house? Well, that's just part of the story. So I hope you understand this in another sense. DNA is what you inherit from the creatures that made you. If we're talking about humans, we're talking about your mom and your dad. You got half of your DNA from your dad, half your DNA from your mom. But all creatures are based on cells, and I want to talk about the different kinds of cells and how they store their DNA before we go into talking about how this DNA actually works. So I'm going to pull up my chalkboard here, well, my grease board, my whiteboard, try not to give you too much glare. And there are 2 kinds of cells in nature. They're the kinds of cells that bacteria have. They're called prokaryotes. And these cells, they store their DNA in little ringlets called plasmids. And these little ringlets of DNA are floating around inside of the cell, and they're kind of loose, left out there. Now, this is only important because of what we're going to talk about with the kinds of cells that humans have. We are not bacteria. We do not have our DNA lying about willy-nilly inside the cell. We keep it in a very special place. So if you've never heard of prokaryotes, you could look that up. Bacteria are the most numerous types of life on Earth, and their cells are all prokaryotic, which means that they keep their DNA in this loose sense. However, let's talk about the kinds of cells that we have. Animals and plants and fungi and all of the big organisms on Earth, we have a kind of cell called a eukaryote. E-U-K-A-R-Y-O-T-E. Now, eukaryote cells, they don't leave their DNA lying about in any place. They have a special structure that you heard that gentleman talking about in that video, the nucleus. That's the nuclear membrane. And the nucleus is the place where the DNA gets stored inside of a cell like ours. It's a special place. And I want to make a metaphor here. I want you to imagine that cells are like towns, or like cities. So just imagine that a eukaryote has a special building for storing the DNA. And the DNA is all the instructions that you need for making another cell. So let's make a metaphor here. Let's pretend that the DNA are like the most precious books. And this nucleus is the town's library where they keep the most precious books. And to help you make that metaphor even more real, I'm going to show you a picture from a medieval town in England called Norwich today. It looks like Norwich. In Vermont, we have a Norwich, but this is the English Norwich. And this is a painting of this town made in the 1500s. And I want you to look at this image and just think of it not as a town, but as a cell. Think of those walls that you see around that town like the cell membrane that stops things from getting in. If they don't want them to get in, they can't get in. That's what the cell membrane does. And then look at those roads throughout the town. Think of those as structures like the endoplasmic reticulum. And I know this is a lot of terminology if you're not familiar with cells, but let's focus on one building in this town that's going to be very important, the library. Inside of this village, let's imagine that there's a library, and in that library are all of the books that you need to build a whole town. It has all the instructions for how to bake bread, all the instructions for how to forge steel, all the instructions for how to conduct a wedding. All these things that happen in town, they have a set of instructions stored in the town library, and that stuff is so precious. We can't just let anybody walk around with it. We keep it in the library. And if you want to come and use it, you can, as long as you're willing to make a copy. You can't take the books out, but you can make a copy. So I know that this sounds silly, but this metaphor hopefully will help you understand how we use DNA inside of a cell. So let me get back from that picture. And so I know that this big little drawing I have here does not in any way resemble that beautiful map of Norwich in England, but let's elaborate on this. Let's make it a little better here. Let's imagine that instead of having a nucleus, we're going to call the nucleus the town library. And here it is. And in the library, we store the sacred copies of DNA. And that library is inside of a larger cell. And the DNA in the library, well, we'll talk a little bit more about how DNA works, but the general idea of how a cell works is that the DNA has the instructions for how to make all of the proteins that make your life work. You can't take the books of DNA out of the library, but you can make a copy. And a copy that comes out of the library comes in the form of another molecule called RNA. RNA and DNA are very similar to each other, but DNA is the double helix, the one that looks like a ladder, 2 strands. And imagine cutting this in half and having only 1 strand, like the bottom half. That's how RNA would look. It's a copy of what's in the DNA, and I'll explain that a little bit more. But that RNA, that gets delivered to the things in the cell that we call the ribosomes. And think of the ribosomes as the baker, the butcher, the candlestick maker, all right, the blacksmith. They have to make the things that work in your cells. And they wait for the RNA instructions to arrive to them, and the RNA instructions are copies of what's in the DNA. So when you talk about this to a biologist, and when you learn about this, in a cellular sense, you'll hear that there's this central dogma. DNA makes RNA, and RNA makes protein. And protein is what makes you work. For example, my muscles and my arms are working because of a protein called actin and another one called titin. And these 2 proteins, they still work, I guess. And they got the instructions for their construction from the DNA in my cells. And that DNA was inherited by me from my parents, my mom and my dad. So let's get a little more into this now. All right, so if you get that book, E.O. Wilson's Life on Earth, I highly recommend it. There are many places where you can go and see what the structure of DNA looks like. I'm going to start by sharing that page we were on, and then moving on to page 12, where I give tribute to Rosalind Franklin. Rosalind Franklin was the X-ray scientist who was able to make this image. She figured out that we knew DNA existed, but nobody knew what it looked like. And she was able to use X-rays to shine through the crystals of DNA. The molecules would be suspended, and then they could shine X-ray light through the molecules, and they could see the shadow, or you could say the silhouette of the molecule in the X-ray light. And it was through the work of Rosalind Franklin that we were able to see that there was this symmetrical pattern. And if I jump from there back to this, you can see how if I put this out in the sun or under a bright light, this is a huge molecule, of course. Remember, the real one is only 2 nanometers wide. So this is the structure that she was able to see. And this was the beginning of humans understanding how DNA works. Not just that it works, but how it works on a mechanical level. Because DNA is pretty cool. It can contract itself into this form when it needs to be stored, like library books that are not going to be read. They get stored like this. And then when a section needs to get read, the entire molecule can unzip itself. And then it actually zips into 2 sections. And that's how the RNA can copy it. So in order for that weird idea to be visualized, let me show you one of the most fun little interactives that you can use in this book on genetics. I'm going to go back to that book by E.O. Wilson, Life on Earth. And on page 15, you can find a really fun little interactive thing where you can zip and unwind DNA. You could while your days away playing with this, just seeing how this molecule works. And if you look carefully, you can see each little sphere is an atom, okay? Atoms like nitrogen and carbon and hydrogen, and they make the molecules, the little tiny subunits that make up this entire DNA molecule. But when DNA is being read, just imagine that that's like the book at the library being opened. Now the RNA can get in and copy the sections that it sees. And when the reading is done and the copying is done, this can be safely stored away in a way that allows it to be preserved. And we'll talk about why that preservation is so important, because what happens if somebody messes up the books in the library? What happens if somebody goes in and takes Harry Potter and the Sorcerer's Stone and turns Voldemort into a good guy? That would change the whole story. And if you change DNA, we call that a mutation. And if I wrote a version of Harry Potter where Voldemort was a good guy, you would say that's a mutant version of that story. That can't be the real story. Well, DNA mutates too. Nature wants some mutations, but not too many mutations, because if you change the letters of a book, the story doesn't make sense. And if you change the letters of your DNA molecule, you could cause horrible things to happen like disease. Some mutations can cause cancer. Other mutations might give you the ability to see better in the dark. So the reason why organisms on Earth have changed over time is because of mutations in DNA. So that's a good thing. But mutations in DNA that are in very vulnerable places can cause deadly disease, death, or cancer. And that's obviously a bad thing. So when we talk about changing DNA, mutating DNA, whether it's good or bad really depends on what changes we're making. So there's some fun with that part of the book. Unzipping DNA. So what is it exactly that we're unzipping? What is it that these little colors represent? Well, the bases, the nucleotide bases, I know that's a big word, are represented here by different colors, but I actually made a little video a few minutes ago with the table behind me that puts some names to these bases. See, they come in pairs, complementary pairs, but this is just a simple little drawing. I'm going to show you their names by pulling up a video that I made a little while ago, and let's see if this helps. All right. So here we have the names of the nucleotides that make the rungs of the ladder of DNA. The G stands for guanine, the C stands for cytosine, the A stands for adenine, and the T stands for thymine. And one of the things about DNA is that these always go together. If there's a G on this side, there's got to be a C on this side. And if there's an A on this side, there's got to be a T on this side. Notice that when we get to this T, there's an A next to it, and this A has a T next to it. And actually, if you want to know, it's a little bit of a joke. Read the side on the left, and I wrote a little sequence. With guanine, adenine, and thymine, I wrote a little phrase. You could say it's gat at a cat. I know that seems silly, but you only have 4 letters to work with, so there's not that many words that you can make. But if on the left, it says gat at a cat, going down the sequence, then notice that the one on the right, it doesn't exactly make that same phrase at all. It doesn't make as much sense. CTA, TAT, GTA. Okay, that is the complementary side to the side that says gat at a cat. So this is how DNA is stored. If there is a mistake on one side, let's say, for example, RNA was getting in here and copying the message, so it was copying the gat at a cat side. Let's say the left side is the side you want to copy, the positive side there. Well, this other side, the CTA, TAT part, what if something happened that while the RNA was copying, it messed things up and it switched these 2 letters? What if it switched the T for the A here? Well, then this side would not fit anymore. When the DNA tried to zip back up like you saw in the textbook, the letters wouldn't fit. The molecules would be incompatible with each other, and then the cell would be able to repair the damage in the DNA. So one of the basic ideas as to why DNA has this double strand is that it's built-in error correction. If there's a mistake on one of these, it won't fit when it comes to zipping it back up, and that will alert the cell that there has been an accidental mutation. So think about a book that could correct itself. Think about a book that if you type the right letters in, it works fine, but if you type in the wrong letters, the book fixes that so that the letters correct themselves. I know it sounds crazy, but that is one of the amazing things about DNA. This is why life relies on this. It's a reliable and sturdy molecule that can self-correct itself if anything is unintentionally or accidentally changed. But it gets changed all the time anyway, even though there's error correction. These changes still happen, and we want them to happen, because if DNA never changed, then life could never mutate. And if life could never mutate, then life could never adapt. If life could never adapt, then I probably wouldn't be here, because I am descended from trillions of creatures that adapted and evolved to live on Earth because their DNA was able to change just enough to allow them to survive, but not too much to make them non-viable, to make them, have a deadly disease or some form of cancer that caused them to die before they could pass on their DNA. So I want to talk a little bit more about this, because at the beginning we saw that video of a cell turning into two. We saw what was essentially every book in the library, every piece of DNA in one cell got copied, and then it got transferred to make a new cell. And just think about that. That's a big job. Imagine if you had to run into the library and copy every single book and make sure you didn't make a mistake. Well, how many letters are in the book? We talked about, I showed you that, that video of the molecule that said "gat at a cat." That's 9 letters long. The DNA of a human being, the genome, as we call it, the genome of the human is 3 billion letters long. So I showed you 9 letters, but there's actually 3 billion letters. And every time that your cells need to copy, let's just think of my fingertip. My fingertip has skin cells, and every day skin cells are dying on the surface of my skin. They flake off and become the dust in my house. That's a gross thought, by the way. But those skin cells that flake off are replaced by new ones that are growing back. So just think about the fact that billions of times a day in my body and in your body, these cells are copying themselves, making a new cell, which means that they have to go into the library and copy every single book, all 3 billion letters of the DNA library. Now, let me show you real quick, instead of just talking about it, let me show you what DNA copying looks like down on the cellular level. That's what's so great about this E.O. Wilson Life on Earth book, is that you can actually see things that would be very hard to explain with words. So here we saw the zipper one more time for fun, good measure. We read the copy and then put it away on the shelf for storage. Well, now let's go to page 20 here, and we're gonna show you a video of how DNA actually gets replicated. And it's pretty incredible because it seems like it's happening so fast, and it has to happen billions of times a day inside your body.
Before a cell can divide in two, it must first duplicate all of its DNA so that it can pass an exact copy to each daughter cell. DNA replication is carried out by a molecular machine called the reprobosome that pulls apart the double helix and makes an exact copy of each strand. The DNA to be copied enters the production line from the left. The blue ring-shaped molecule is the enzyme helicase, which unwinds the double helix into 2 strands. The leading strand is copied continuously. The lagging strand runs in the opposite direction with a more complicated copying mechanism. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Now think about that. Hold on a second. That has to happen billions of times every single day. 3 billion letters copied, hopefully perfectly, but actually, it turns out that that amazing system with the helicase and the reprobosome, all of that copying of the DNA, on average creates 1 error per billion base pairs. So that means every time a cell divides into 2, there's going to be on average 3 or so errors in the new DNA that those cells have from the old DNA. So there is the opportunity for mutation. Chances are those 3 mutations will not actually affect much. There are lots of places in the DNA. You could say that they're like the blank parts of the book, where if you make a change, nothing's going to happen, like on the margins or on the blank pages at the beginning and at the end of the book. But if you change the letters in the part that's very important, it can have deadly consequences, and I'm going to go into that. But I wanted to go back again to that video that I showed you, because there was actually information being conveyed in those random letters. When I chose "gat at a cat," I was choosing that because it sounded like something that's easy to remember. Gat at a cat. It sounds funny. But those letters actually code for something in your body that will eventually become a protein. And think of those little enzymes, those little machine-like molecules that were taking the DNA and copying it and reproducing it. Those are proteins. Those are enzymes. Those are the kinds of things that your DNA knows how to make. So those actually little machine-like molecules were made from the information in your DNA. And if I go back to the video that I made earlier with the letters, gat at a cat, well, it turns out that DNA, GAT, those three letters put together actually call for a recipe. They're like an instruction for your cell to gather up a molecule called aspartic acid. And then the ATA, the ATA part, on the left, A, T, and A. Oh, hold on, I'm sorry, this is getting a little, let's see, we got the GAT, that calls for aspartic acid. We got the ATA, that actually calls for isoleucine. That's another amino acid. And then if I go down to the last part, CAT, C-A-T, that actually calls for histidine, a third amino acid. So I know GAT at a cat sounds like a silly number of letters to you, but inside of your cell, that GAT codes for a specific amino acid that is needed for a protein. The ATA codes for a different one. So the ATA stands for something else, and that is the isoleucine. And then the CAT calls for the ingredient histidine. So maybe thinking of recipes is a good way to think of this. Your DNA is the recipe. It's not the cake. It tells you how many eggs, how many cups of flour, how many cups of sugar that you need. It doesn't look like the cake, just like a cookbook doesn't look like food, but the cookbook has all the information that you need to get the food made. So the DNA is the recipe, the RNA is the copy of the recipe, and the cake or the product at the end is a protein. So DNA makes RNA, RNA makes protein. That's the simplest way I can summarize this. But let me give you a very specific example. That gat at a cat, that didn't really code for much of a protein with 3 amino acids. It's not going to do much. But here I have something that was printed out by a company to show you how long in letters this would be. So do you see the letters there? A, C, A, T, 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 G, C, T. This is the actual code for the beta globin gene. This is something that's on one of the chromosomes inside of your cells. You have 23, pairs of chromosomes. On chromosome number 11, on that stack in the library, you could consider the chromosomes like the shelves in the library, there is a beta globin gene. Now, if you don't know what beta globin is, maybe you've heard of hemoglobin. Hemoglobin is the protein that's inside of your red blood cells. When you breathe in, you take oxygen into your lungs. The hemoglobin in your red blood cells has iron in it. This is why your blood is red. This is why when you lose a tooth or cut your tongue, it tastes like rust. Hemoglobin has a lot of iron in it. Iron is what makes the oxygen stick to your red blood cells. So hemoglobin is a complicated protein made of alpha globin and beta globin. And this is just the recipe for beta globin, by the way. And every single one of your red blood cells has this protein in abundance. This is why oxygen from the outside air sticks to the red blood cells in your lungs. So let's think about that very specific recipe. We need this protein to make oxygen stick to our cells so that we can live. Is it important? Well, of course it is. This is the gene that covers how you get oxygen. So if this gets messed up, you might not be able to get oxygen from the air, and that means you won't be able to survive. But look at how small these letters are, ACA, TTT, GCT, and look at how long this ribbon has to be. I mean, do you want me to read them out to you? Of course, it would take forever. This particular protein requires 147 amino acids. So every 3 of these letters, like the first one, ACA or TTT, they code for a different amino acid. They're called codons. I don't want to overwhelm you kids with all this terminology, but this is how DNA is read. It is letters that, well, it's not really letters, it's actually molecules that code for amino acids, and those amino acids make your proteins. So here's the protein that helps you breathe. And look how much information it takes just to code for that one protein. And just think about the fact that that is one little section of the DNA inside of your nucleus town library stored away. So if that DNA got messed up, your body would not be able to make globin, and that means that your body would not be able to absorb oxygen from the atmosphere. Obviously, bad news. So I hope that this idea of how something encoded in all these letters that actually represent different molecules, the nucleotide base pairs, turns into something in your cells that helps you live. That's what DNA does. And just imagine, this is one small section. You have 3 billion letters of that code. Now, see how hard it is to store that stuff in a sensible way? It's making a mess over here. Maybe I should follow nature's example. Instead of letting this pile up in a big noodly mess, is there a more organized way for me to put this away? Sure. Remember how that DNA was wrapped around all those nucleosomes like this? Like a garden hose on a reel? Well, wouldn't that make it more convenient for me to store this information here too? Maybe if I stored it like this, it would take up less space and it would be safer, less likely to get damaged. So now that looks better. And that is how DNA gets stored inside your chromosomes. It's a little more complicated than rolling up this tube, but pretty soon, that big bunch of messy paper that was hard to control and hard to keep in a nice pile, well, now it's much safer, more stable, and easier to transport. That's how your cells deal with DNA, sort of like going from this to this. So now, at any moment, we're about halfway through our lesson, folks, and I've just gotten through some of how DNA works in a mechanical sense. But if there's any questions, I hope there are, please let me know if anyone has any questions they'd like to ask. Maybe this doesn't make sense yet. If there's something I can do to help, please let me know. But now, let's move to the next step in this process. We talked about how DNA stored in the nucleus, the town library. Maybe I should write that up here. Okay, then the DNA is unfolded, copied, and folded back up in the library. It stays in there. We're not a bacteria that lets our books from the library sit out on the sidewalks all over the place. We keep them organized in the library. But the RNA goes out, and that RNA gives the instructions to how to make those proteins to the ribosomes. So let me actually show you how RNA copies off of DNA. This is kind of like the same thing as if I were to show you in the library how to take the encyclopedia and stick it on the photocopier and press copy, so you get a piece of paper that has the exact same information as the encyclopedia, but then you take the encyclopedia, you fold it back up, and you put it on the shelf. If you can understand that metaphor, then get ready for this. On page 24 of that book, Life on Earth by E.O. Wilson, we have RNA polymerase. That is the name of the protein, the enzyme that actually copies the DNA and turns it into RNA. And the RNA can then leave the nucleus. So let's let that play.
Transcription is the first step of turning a gene into a protein, in which the genetic code is transcribed from DNA into RNA. The protein enzyme RNA polymerase transcribes the genetic code in DNA, letter by letter, into an RNA copy. This mechanism for RNA transcription is presented in real time at around 30 bases per second.
30 bases a second. Okay. Did you ever think a copying machine would look so cool? I mean, just think of how it's churning through that DNA to copy and Then once you have that RNA copy, then the RNA can be sent out into the ribosomes, and that's where the magic of making proteins happens. So we're gonna skip to the next step. We just saw DNA turn into RNA. And did you notice, in case you weren't paying attention to the visual part of it, the DNA looked like this, the double helix. But RNA is always just the single strand, the half that is necessary for the information. So remember, the general sense is that one half of this has the information, the other half is that mirror image backup copy that prevents this half from getting damaged. So RNA doesn't have that backup. RNA is a single strand, so it is more vulnerable, but the good thing about RNA is that it only needs to last for minutes to hours while it gets used. It's just like a photocopy. Once you've gotten the information and you've baked the cake, maybe you recycle the recipe and throw it in the waste bin or the recycling bin, I bet. And then you don't need it again until it's time to make another cake, and then you go get a new fresh photocopy from the nucleus. So let's go imagine that step, turning the photocopy, the RNA, into a protein, like a recipe into baking a cake. We're gonna move to that, and that's gonna be on page 30 of the E.O. Wilson Life on Earth book. So I'm gonna jump to that page now. We're going to meet a new kind of RNA.
The ribosome is a large molecular factory that translates the genetic code in messenger RNA into long chains of amino acids that fold up into a protein. The gene code is read in groups of 3 letters called a codon. Amino acids are delivered to the ribosome by transfer RNA and match to the gene code in the messenger RNA. Once gene translation is complete, the amino acid chain folds up to create a protein with a 3-dimensional shape that determines its function.
Okay, now wait a second. This got confusing. Now we just heard of 2 kinds of RNA. There was messenger RNA. That's the one I've been talking about, the photocopy. And then there's transfer RNA, and I have a little model of transfer RNA. You may have noticed they look like little tiny boomerangs. And they basically have on them the 3-letter code that needs to match up with that particular part of the code. So basically, this is like a reader and a depositor. This is, let's pretend that this is the one that's looking for the code GAT. At the beginning of my code, the one I put on the table, GAT, that calls for aspartic acid. Let's say that this actually has the aspartic acid on it. So on this side, instead of having GAT, this would have CTA. This would be the complement for the GAT. And it says, I'm looking for GAT. I've got CTA. I match with GAT. And if it matches, it says, aha, I'm in the right spot. Then it deposits the correct amino acid. And a protein is basically a bunch of amino acids put together. So think of each amino acid as a little train car, and the protein is the train made of all the little train cars. And then the last step, which is something that could be a whole class unto itself, is how the protein folds up on itself. After the protein train is built with all the little cars, then the protein can change into a 3-dimensional shape, like that replicase that splits apart that DNA. In fact, proteins and enzymes are fascinating. They're really complicated things, but their form is based on which amino acids made them and how they fold up after they're created by the ribosome. So just think about that. All these proteins in my jaw muscles, making my body work, making me speak. The diaphragm muscles in here, they're all proteins. They're all my body's muscles, all my motion, all my functions are regulated by these proteins. These proteins were made by the instructions in the RNA, and the RNA got the instructions from a copy from the DNA. Whew. I know it sounds complicated. There's a lot of words, a lot of molecules, you know, like guanine, cytosine. If you focus on that part, you might get confused. But if you focus on the structural stuff, how it works, how the information gets passed from one layer to the next, then it's actually not that complicated. Don't let the words and the big terms scare you. Let those diagrams, let the textbooks like Life on Earth by E.O. Wilson, let them guide you, because that can help you get through the terminology. But just think of the basic concept. It's not that complicated. It's copying information and then turning it into something that you can use. But let's focus a little bit now on the things that can happen when things go wrong. We call this mutation. Like I said before, some mutations can give you superpowers. Oh, I'm Wolverine now. I'm an X-Men. No, just kidding. Sorry, kids. Mutants in comic books are different from mutants in real life. A mutation, for example, can cause you to become an albino, where you don't have the protein called melanin that gives you the pigment that makes your hair. So you'll have very light color hair. And if you don't have pigment in your eyes, you could have pink eyes. Why? Because an albino doesn't have the protein melanin in a functioning way. That's the kind of mutation I'm talking about. So let me talk about a very specific mutation that is talked about on page 34 of the E.O. Wilson book. I'm going to skip ahead to the next section, which is all about mutation. And here is a perfect example right on the page of a normal and a mutant DNA sequence. Look very carefully where it says under normal, CCT, GAG, GAG. And then look at the mutant side where it says CCT, GTG, GAG. That is literally one letter difference. The adenine got swapped for a thymine in the DNA, and it's going to have enormous consequences for whoever has this in their DNA. Because when that sequence, by the way, this is the globin gene, the same gene that we were talking about with my long ribbon of paper that I rolled up, that same gene can be changed in one place, one letter, and that will cause somebody to have a very different type of red blood cell. So if you look under the normal column, that DNA sequence leads to a protein that has glutamate. That's what GAG codes for. That's the amino acid. And then that protein in the cytoplasm helps to build that nice looking red blood cell. And by the way, those red blood cells, they look like bagels or donuts. Those are the ones that make your blood red. Those are the ones that are rich in iron, that they absorb the oxygen from your lungs. So they are the ones that hemoglobin works in. And if you make the mutant strain, if you look at the DNA here with one letter changed, you'll see that instead of calling for glutamate, it'll call for valine, a different amino acid, and that causes the same protein to make a different shape. And that shape of the globin protein causes the red blood cells that are filled with that protein to become sickle shaped. So those of you who are out there listening, maybe you're familiar with sickle cell anemia or sickle cell disease. This is a very serious problem that affects millions of people. By having this one simple change in their DNA, they have a lot of red blood cells that are misshapen. And so when they're shaped like a sickle, they're more likely to get caught in the blood vessels. They're more likely to get cause clogs in your arteries and your veins. And that can cause enormous health problems. It can cause people to have low energy, the inability to do anything like even go up the stairs, and it can cause death. However, there are many people who have this mutation, and it doesn't, it doesn't kill them. It just makes their life a lot harder. So they could go on and have children, but that means that when they have children, those children are inheriting this DNA that might make their lives more difficult and more painful. That is what sickle cell disease does. So it's a mutation that's bad enough to harm you, but not bad enough to kill you necessarily. So many people have it and live to be adults, and that's why they are able to pass it on to their children. So there's a lot of people with this gene. But let me put the other side of this. If you have the sickle cell gene, you also are less likely to be stricken with malaria. And malaria is a horrible disease that kills about 1 million people a year. So think about that right there. Let's think about that. That mutation causes pain and discomfort and shortened life and difficulty in life for people, but it also gives some protection to the most deadly killer of humans in all of history, malaria. It's thought that about half of all humans in history, going back to prehistoric times, have died from malaria. So is that mutation a good thing or a bad thing? That is a difficult question to answer. It has benefits and it has detriments. It has bad side and a good side. For some people, the bad side is so bad that the good side isn't worth it anymore. And one day, let's hope that humans are rid of malaria. Let's hope that that disease can be, wiped off of the face of the earth, and humans will never have malaria. So then you would say, this gene is only bad. Nobody needs the malaria protection. And all those people who are suffering through all that discomfort, how can we help them? How can we fix this mutation to stop a person from suffering from sickle cell disease? Well, now you understand why scientists are looking into how to manipulate DNA in ways to help people. Gene therapy is one of the names that it gets called. When you find out that there are some diseases that are caused by bad DNA or mutations in the DNA, naturally the next question is, can we fix the DNA and then fix the disease? And there are thousands of diseases that are caused by mistakes in the DNA. And let's remember that cancer is caused by mutations in DNA. So anything that you could do that could fix a broken piece of DNA could cure thousands of diseases and cure cancer. But then what about changing the DNA in other ways instead of just fixing mistakes? What if I decided to change my DNA and give myself the ability to glow in the dark? I know that's not a very practical thing to have, but is that also possible? Yes, it is. But that would not be the same as fixing a disease. That would be more like modifying myself to enhance or augment myself. Like if I was trying to give myself superpowers by changing my DNA, that would be a very different use than changing DNA to fix a disease. So let me actually show you a news article. Actually, it was a news report from National Public Radio about a person who has sickle cell disease. Her name is Victoria Gray, and she lives in Mississippi. And she's a mom. And as her kids are growing up, this sickle cell disease was making it harder for her to keep up with daily life and be able to be a part of her kids' life. She would have to sometimes spend days in bed because of the complications caused by sickle cell disease. So she decided to volunteer for an experimental project where they were going to change her DNA. I'm not going to go into all the details. Something called CRISPR is involved, and that's a new process that scientists have figured out that allows us to change DNA more easily. But let's just say that Victoria Gray is a very brave volunteer. I'm going to show you the article from National Public Radio so you can listen to the story sometime. Oh, hold on a second. That's a different article from the New York Times. But here is Ms. Gray, Victoria Gray, and she volunteered to have her bone marrow DNA changed. Now, just let me get some things out of the way. Ms. Gray already had her children, so any changes that happen to her DNA now will not affect her children who are already born. But also, just think about the fact that the DNA that they're changing is in her bone marrow, which is where red blood cells are made, and that's where you need to do it if you want to fix the red blood cells that cause sickle cell disease. But if you wanted to make this change in her DNA pass on to another generation, this would be like a question. I don't think we're going to have anybody answer it today, but they would have to change the DNA where? Where would they have to change the DNA for it to actually affect future generations? Not in her bone marrow, but let's imagine that Ms. Gray chose to have another child. They would have to change the DNA inside of her egg cells in order for that change to pass on to another generation. So I just want to be very clear that what they've done to Ms. Gray is not going to affect any other human being. It doesn't have the ability to pass on like your parents' DNA passed on to you, because that process is mediated by the cells we call egg and sperm cells. So they did not change all of Ms. Gray's body. They just changed the DNA inside of her bone marrow, which is where her red blood cells are generated. And I'm not going to go into the whole details of the article, but this is an amazing story. She was in the hospital for months, and I want to just jump forward a little bit to show you a picture of sickle cells themselves. So in the article, you can see red blood cells that are normal and healthy next to the sickle cells that you could tell by their blade-like, sword-like shape that they're going to get caught in places. They're not going to flow as easily through a blood vessel as those round ones are. So they basically figured out a way to modify her DNA, and through many hospitals, through a CRISPR process that changed her DNA, they took out her old bone marrow that had the messed up DNA, and they replaced it with new cells that had this corrected DNA, or you could say, improved DNA. And now Ms. Gray is able to participate in the daily life of her kids. She's already back home. She's able to climb stairs and go to football games and do things that she used to not be able to do because of her disease. So if you listen to the story, you'll hear Ms. Gray at the end say, "I'm a GMO." She called herself a GMO, a genetically modified organism. And that is what I want to get to next, GMO. Today, let me see if I can erase this little silly drawing here. Today, if you ever go shopping for food, you might actually see things that say GMO free, meaning it does not contain GMOs. GMO stands for genetically modified organism, meaning something that we changed the genes of, modified, and a lot of people think that there's something wrong with GMO food, so you'll see a lot of foods that say GMO free on them, and you'll also see things that say produced with genetic engineering. Now, these terms do sound scary to some people, I can imagine, but what does this really mean? Genetic engineering, genetic modification. This is mutations done on purpose. And for a lot of folks, they think this is something totally new that has come from recent laboratory science. Many people think of things like Jurassic Park or Jurassic World, where they took the Velociraptor and the Tyrannosaurus and made the Indominus Rex and all those things. No. We're not talking about that. We're talking about how GMOs are already a part of life. Now, this is where some people might be surprised. Humans have been modifying the genes of organisms for thousands of years. We used to call it breeding, when people would take one organism and breed it or change it to become another. For example, a cow. Cows don't exist in the wild. They only exist on farms. And back in Europe, thousands of years ago, there was a giant wild cow called the aurochs. And the aurochs are now extinct. They're gone. But long before the aurochs disappeared, some clever humans captured some of them, said, "Oh, mommy, can we keep it? It's so cute, this little calf." And they took it home. They controlled how it bred. They picked its mates, and they modified it over time, not by going into the cell and changing the DNA in a laboratory, but by saying, "Hey, that bull and that cow should get together and make a calf, and we'll see what the results are." And that's how we ended up with Jerseys and Holsteins and Ayrshires and Herefords and Black Angus. All of those different kinds of cattle are descended from what was a wild creature that no longer exists. So could I say that a Jersey cow is a genetically modified aurochs? Yeah, that's completely true. But how it was modified was by animal breeders picking mates, males and females. Let's put them together and see what happens. So let me give you another example that you may not know as well. Let's talk about how we got to corn. So hold on a second. I'm going to switch to a little presentation here. Here is a plant that is a wild grass from Mexico and Central America. It's called teosinte. If you look at this grass in the wild, you would never imagine that this could one day become the most important food crop on Earth. This happened thousands of years ago. This happened in prehistoric times. We don't know who did this, but we do know that it took generations of farmers in Mexico, Native American cultures, taking this grass, controlling where the pollen went in the wind, and figuring out which ones they wanted to cause to reproduce. And eventually, this little grass became what we now call corn or maize. So look at it on the left. How did that little thing turn into that big corn on the cob on the right that looks so delicious? I can't wait to put some butter and pepper on that thing. That looks amazing. And you would never have guessed that you could turn that little grass on the left into something that big. But how was it done? It wasn't done in one step where somebody went into a cell and changed all the DNA and got that corn. It took time. It took generations of plant growers, farmers saying, "Woo, that one has bigger seeds than the rest. I'm going to save those seeds, and instead of eating them, I'm going to plant those seeds next year. And that's when they made a choice that caused the genes to be modified. They picked the genes that they liked, they ate the seeds that they didn't like, and they kept the seeds that they did and grew them again. And over time, thousands of years later, we got to what we now call corn. So this was done by Native American cultures, mostly in the Oaxaca region of Mexico in ancient times. This is genetic modification, old school version. It takes a long time, and you don't know who to give credit to because it's actually the work of thousands of farmers over countless generations. All right, well, I'll show you another picture. So there's teosinte on the left, there's maize in the middle, and you can see the corn cob from both of them next to each other on the right. And I would bet you would never think teosinte could have turned into maize, or just imagine how long that would have taken by natural mutations, accidental DNA copying errors. Maybe it could have happened too, but that might have taken millions of years to happen by natural mutations, maybe thousands of years by humans intervening by controlling breeding. But what if you wanted to do this in not thousands of years, but in just a few years? So when we hear about GMOs in today's world, we're not talking about old-fashioned plant breeding. We're talking about modern methods where they manipulate DNA to create a completely different organism. And I'm going to put up a real quick diagram, although I don't expect you to be able to read all of this at once. This is sort of the basics of how GMO works. You find a gene that you want in your plant, you get that gene out and you copy it and you try to introduce it into a new organism. And there are many ways to introduce it, but interestingly enough, I found out that one of the methods that they use in modifying plants is to use a virus. There's a virus, tobacco mosaic virus, that if you change its DNA, you can get it to do the DNA changing for you. So humans didn't really invent this technology. We're using things that have already existed in nature, like viruses, to modify the genes of these plants. There's also bacteria that, through their normal survival, modify the DNA of plants that they try to live on. So there are already bacteria that figured out how to genetically modify organisms before we did. And scientists have identified these bacteria and these viruses, and then basically we use them. Think of like the Trojan horse. We use them as a way to get that new DNA into the new organism, and then we grow that new modified organism to see if the DNA characteristics that we wanted actually turn out in the creature. And I want to toss this over to a professor. I'm not going to let her speak, but there's actually a professor who is involved in this kind of work that works at Dartmouth College. So I'm going to end it with this because I want to give a face to GMO. And here's Professor Mary Lou Garino. She works down at Dartmouth College, and what she does is she tries to figure out how to take crops that people grow around the world and make them better by having more vitamins and minerals inside of them. For example, let me give you, here's a picture of her the work from her lab. And actually that picture on the right is a leaf where all the metals like iron have been mapped out. So you can see where the metals travel through a leaf. And that is her job. She tries to figure out how metals are absorbed from the soil through the roots and how they work their way into a plant's leaves and tubers or seeds or nuts. And if you were worried about people's nutrition, you would want to know, how could we get this crop to have more iron? Iron deficiency causes anemia, that's a disease, and that affects millions of people around the world. What if we could grow a crop that in the same field gets more iron out of the soil, and then when people eat it, they get the iron that they need and their nutrient deficiencies go away? We could be curing lots of diseases by making these plants have more iron in them. So just to quickly wrap this up, as our time together is almost at its end, I'm going to show you just a little example of some of the work that Professor Garino has done. This is the plant that she has used as her study subject. It's the model organism for her genetic modifications. It's called Thailcrest, but most scientists just call it by its genus Arabidopsis. Now, through studying this plant, she was able to understand how iron is absorbed from the soil and how it migrates to the leaves and the flowers and the stems of a plant. Then she applied that knowledge to another plant. This is the Arabidopsis, the plant that Professor Garino uses in her studies. But the target of her research was another crop that I know very well. This is something that some people call cassava. In the Latin American world, people call it yuca. It's also known as manioc, and it looks like gigantic potatoes. This is a very important food, and I know because I grew up eating it. Cuban culture and folks in the Caribbean love to eat yuca. You kids may not have eaten yuca in that form, but you've probably had tapioca pudding, which is also made from the same root plant. Where I grew up, I eat it as yuca frita, fried yuca, a very delicious snack. That is a plant that a lot of people in the world eat, and in many places where there's a lot of poverty, this plant might be the only affordable source of food. So what Dr. Garino was able to do was to find a way to get this plant to grow, the cassava, but she modified its genes so that it absorbs 10 times more iron from the soil than the normal yuca, the normal cassava does. So she is making a GMO, but her purpose is to improve human health. And is her work really that different from what an ancient farmer in Mexico was doing when they tried to get seeds that were a little bigger, a little more delicious for their corn? So I just want you folks to know, GMOs are a part of our lives today. You may not like the idea that scientists can change DNA, but just think about the fact that humans have been doing that by other means for thousands of years. There are other ethical problems with genetically modified crops that have to do with intellectual property and copyright, and I'm not going to go into all of that, but I'll leave it with this because I want you to know that I found an article from Time magazine that's already 5 years old, but on this article, it lists the amounts of foods that you already eat that are genetically modified. So this is 5 years ago, and the numbers have increased by a lot, but soybeans, cotton, corn, some of the clothes you're wearing might have been grown with genetically modified cotton. And the corn in your cereal and in your chips almost certainly was genetically modified. The soybeans that feed our cattle and our pigs and our chickens are also often genetically modified. So you can see beets, squashes, even apples, tobacco, who we don't really need that. But the point is that this article will show you how many different companies have already modified crops. Are all of them doing it for the benefit of humans, like to get the crops to be better food with more nutrients? Not necessarily. Some of them are doing it just to make their crops more profitable, and that's where the ethical issues might come in. But some of these are modified to be resistant to insects. Some of them are modified to be resistant to pesticides or herbicides. So you can spray the crops and the plants won't be damaged. So the point is, GMOs have been part of human life since we made dogs out of wolves and made cows out of aurochs. Whether the GMOs of today being made in a laboratory are that different from the ones made by a farmer in the field, that's a different question. But I hope you understand that regardless of how you feel about the GMO issue, what we're talking about when we're talking about genes, the G stands for the genes in your DNA. So hopefully now you can understand how DNA works, and I hope you get that E.O. Wilson book, Life on Earth. Unit 4 is the one about genetics that will have all those diagrams and videos that I showed you. And if that doesn't get you, then I hope you continue to study this subject and just pay attention to the news and read the labels on your food and see how you interact in this world with genetics and gene modification being a part of our daily life. So do we have any questions on there, Drew, from all the folks?
Oh, I think it's a little bit quiet, but we're a few minutes over, so I just wanted to thank everybody for joining us who is in our Zoom, our YouTube, or might watch this later on cable access television. We've been really excited to have you with us today. And a warm thank you, of course, to Bobby Farlice-Rubio, our educator on genes, mutants, and GMOs. And we'll see you soon. And if you're looking for other online programming for your school day, of course, check out fairbanksmuseum.org, and you'll be able to find a link there to all of the different online stuff that we offer. Thanks again, Bobby, and have a good day, everyone.
All right. Thanks, everyone. Stay well.